All right, SBI4U. This is video number 6. It's our Monday video, so welcome to a second week of virtual learning. This is called Applications in Photosynthesis. So it's just a quick one about different things that we can do to kind of play around with how plants grow and how they photosynthesize, and also different adaptations that plants have made, because they can't move and they can't move themselves to a different place and adapt that way, but they can adapt in many other ways. So we'll talk about that. Mostly this is going to be based on the stuff we already covered about like wavelengths and the Kelvin cycle and things like that. So that's what I'm sticking to in terms of applications. We're not going to talk about like fertilizers or anything like that. So applications of photosynthesis. What we're going to talk about here, first off, is the spectrum of photosynthesis and grow lights and choosing a good grow light and stuff. We'll talk about actually directing a plant's growth with light. So different light will actually make different plants do different things. Then we'll jump over to the carbon side of things. So different plants actually have different ways to manage their carbon levels, especially the carbon dioxide. And then we're going to talk about RuBisCO, that enzyme, the #1 protein, the most popular protein on the planet, and how it actually is not super efficient, and we'll fix it. People are working on fixing that with genetically modified organisms, genetically modified super plants, basically. First off, just real quick, space lettuce. They grew lettuce on the space station, and I'll mention that in our first bit here, which is grow lights and wavelengths. So grow lights and light spectra. Here we go. I'll stick my my face down here while we do that. Grow lights and light spectra. So first off, here's once again the chlorophyll A and B absorption spectrums. Chlorophyll A absorbs, kind of further out than chlorophyll B. The specific numbers are up on top here. 680 nanometers and 700. That's the reaction centers that we learned about yesterday in the on the electron transport chain of the plants in photosystem 2 and photosystem 1. Photosystem 2, 680 nanometers. Photosystem 1, 700 nanometers. But those are both over here in the red. Over here in the red. Here's a few different light sources, common light sources, and we can kind of compare how well they are at hitting the 680 and 700. So sunlight is very good. It's a very broad spectrum thing. So yeah, that's great. A tungsten lamp, like a standard incandescent light bulb, basically, one of those old school light bulbs, is very good in the red, but not very good in the blue. A white LED is very good in the blue, but not very good in the red. And a mercury vapor lamp, like those fluorescent tubes that you see in office buildings, and those little light bulbs that have like the spirally pattern that are basically tiny versions of those mercury vapor lamps, those are terrible in the red, really, really good in the green and kind of the blue over here. It's actually really interesting if you look at those, if you bounce them off of like a CD or your phone screen, sometimes even, you'll get specific dots of color instead of a spectrum like you normally get when things reflect off of a CD, because it's only hitting these wavelengths. Kind of a fun little at-home science trick you can try. Maybe I'll put up like a picture of that on SBI or something like that. You guys can see it. Yep, so that's that. And then the barcode scanning laser is very, very, very specifically 650 nanometers for the wavelength. So if we think about 680 and 700, 650 is not going to cut that either. So you can't grow your plants with a laser. I'm sorry. Yeah, so normal light bulbs don't do it very well, but grow lights do. So here we've got a blue peak, and a red peak. This would be like a typical grow light. So a grow light will specifically try to peak in blue and red because that's where the chlorophyll likes to absorb. I might actually pause for a second and see if I could do an experiment with my aquarium light and see if you can actually see that blue peak on it. So give me a second, I'm going to pause it. Okay, so this is my fish tank. I've got the lights on full brightness. Oh, that's the timer. Nice timing, eh? Okay, let's get that back going. Okay, so the lights are on full brightness, and I've got a CD here, and you can see, you know, it's shiny. But if I get the CD close, these lights are actually like a bunch of individual LEDs. If I get in real close with the CD and I angle it towards the fish tank, somehow, there we go. You can see the here, just here on the disc right there, you see how there's like a reflection of the spread of the LEDs? So it's hard to give you guys an accurate picture of that right now, but I took a better picture of it, and I'll put it up on screen. Just give me a sec to pause. Okay, so this is what it looked like. I took a picture with my phone of me holding the CD right up to the light. So the light is up here. You see it bouncing off of here. And this is those reflections I was talking about. Each individual LED has its own little reflections. That's why they're in rows of 3. 1, 2, 3. So those are all coming off of there. And I want to highlight this little blue right here. Like, look at how much blue. That one extremely bright blue spike tells you that LED, that they've chosen LEDs that are very, 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 very spiky in the blue, just as we expect. And here's kind of a closer in view, and you really do see that blue spike, and some red, yeah, a nice red spike too. The yellow and green are there just so it looks, you know, nicer to our eyes instead of looking super purple. So yeah, that's wavelengths. It's very, very cool. You can do that yourself if you have one of those CFL light bulbs, those like spirally ones, and a CD. You'll see very specific bands off of the CD. Just hold it at kind of like an angle like this, and you're looking at it kind of like that, and you should be able to see okay. It also helps if you make a little pinhole for the light to go through instead of having the whole light bulb. Just a little pinhole helps a lot if you want to try that out. All right, back to this. So yeah, they can actually you can actually get away with just red and blue if you don't care at all about what your setup looks like. Obviously, the fish tank, they want it nice and white so that the rest of it looks good. But theoretically, you can just do red and blue because that's what the chlorophyll needs. And that makes it more energy efficient. So here's the space lettuce. Notice how they grew it, right? They grew it in just red and blue light, just red and the purple is just going to be a mix of red and blue, just LEDs, because they don't need the green. Why would they? No. And so they were able to make lettuce in space and eat it too. They actually had a salad that was made with the lettuce that they ate. Yeah. That's that. I'm going to pause while I set up the next thing. Okay, next up, our next topic is photomorphology, which is just a fancy way of saying that the light of a plant will affect its growth patterns. So take that away. And that says growth patterns, so if I put this over here, growth patterns. But I'm going to stick up here because that's an easier place to be. What you can see here is that these were some snapdragons that were grown under different kinds of light. Here's blue, red, and far red. So like really, really, really close to infrared, but you can still tell that it's red. So blue, red, and far red. You can see that all of these have far red, and all of these have flowers. These have flowers. This one had no far red at all. No flowers. It's sad. The reason for this is that plants use the different kinds of light that hit them as signals for what's going on outside. So like far red happens a lot at sunset. Sunsets are very far in the red. So at sunset, that kicks in the plant's kind of night cycle, and part of that involves flowering. Also, the amount of far red tells it that it's spring or winter. Like they actually keep track of how much is hitting them over time, and it tells them when to flower and when not to flower. So if you don't put a lot of far red in, the thing won't flower, or it'll make really tiny flowers, stuff like that. Like here, for example, this only had 16 of the far red, and it has smaller flowers than the other ones that had more of it, and stuff like that. Red light promotes leaf growth. So you see that red, this is 128 red, lots of leaf growth. 96 red, not as much leaf growth. 64 red, also really sad leaf growth. 128 though, pretty normal leaf growth. And if you look at the leaves here, these leaves are big and luscious because it's all red. These ones got less red, so not so great. This had a lot of far red, so I guess that helped out for the leaf growth. And the rest of them have pretty normal looking leaves. You can see the far red 32 versus far red 64 is the only difference here. And you got, you know, squatter plants with nicer flowers. Yeah, so that's far red. And then here's another experiment that they did where all red and some green, you know, red, some green and some blue, red, all blue, stuff like that. And you can see that the one that was in all red has the longer kind of stems and the bigger leaves. And other ones are squatter and stuff like that. So yeah, far red promotes growth. The reason for that is far red will penetrate through shade, and it'll even penetrate into the ground a certain distance where the seeds are. And it's what sets off the stem to make it grow higher. So you see that these stems are really tall because it's got a bunch of far red. So are these compared to these. So that's part of it. And far red also promotes flowering, like I said. And one thing that you want to flower if you're growing it indoors is marijuana. The cannabis plant, the buds that contain the THC are, you know, part of the flowers. And so this is actually a picture someone took. It's not my picture, but it's a picture someone took of what the horizon looks like in the east end of Ottawa these days out near Trim, because there is a commercial cannabis grower. And I live in the east end of Ottawa, and I see this. I see this on certain cloudy days when it's evening. Their grow lights, because they're using so much far red, it looks a lot like a sunset, because that's what they're trying to do. They're trying to tell the plants, hey, it's sunset. You know, it's long sunsets, so it's springtime. Make yourself some flowers. That's what they're trying to show their plants. And they're trying to use photomorphology, the current topic, to affect how fast their plants are making flowers, to make more marijuana faster. So it's kind of cool. Also a bit of a nuisance because it trips me out every time. It really does look like a sunset on the horizon, except it's on the wrong place. It's on the east side instead of the west side. Yeah, it's neat. All right, I'm going to set up the next topic, and the next topic is going to be carbon cycles, different kinds of carbon cycles for plants. So we'll pause. And we're back. So we're going on to C3 versus C4 versus CAM. These are different carbon cycles, so just jump right into it. The keep in mind here is that all of these have a Calvin cycle. So here's the Calvin cycle in the bundle sheath cell, which is all plants have that. The difference is some extra cycles that certain types of plants can add in to kind of separate the CO2 piece from the Calvin cycle piece, because if it's just a Calvin cycle, CO2 has to come straight in from the stomata and go right into the cell. And that means you can only do the synthesis part if the CO2 is actively coming in. You can only do it during the day. You have to have your stomata open during the day when you're getting hit with sunlight and it's warm, so you're going to lose a bunch of water. So if the plant wants to lose less water, there's two strategies. There's two strategies to prevent drying out. So this, for example, rice and wheat and stuff like that are C3 plants. I believe wheat's a C3 plant. Let me just double check that before I make a bad statement. Okay, thanks to the power of Google, I now know that wheat is a C3 plant. So about 80% of plants on Earth are C3 plants. You see C4 and CAM plants in drier places because there's an evolutionary drive to figure out how to prevent the water loss from the leaves. They've worked out some interesting ways to do it. C4, a lot like C3. I should move this to the bottom corner. C4 is a lot like C3. It's still just a carbon cycle. There's no like other thing, but there's this extra cell here, the mesophyll cell, that takes in the carbon dioxide and runs it through a separate carbon cycle. And then it doesn't pass carbon dioxide as a gas to the bundle sheath cell. It passes this C4 molecule that drops off the CO2 inside the cell and then comes back as a C3. So it separates the intake from the air from passing it to the bundle sheath cell. This is important because the water is being used over here, so you end up with kind of like an insulation layer, like an insulating layer out here. And it reduces the loss of water because the water is separate from where the CO2 has to come in. So that's how C4 does it. Corn, maize, corn. The corn plant is an example of a C4 plant. So it can grow in much drier climates, much hotter and drier climates than rice can. If you've ever seen a rice field, rice fields are just completely inundated in water because rice needs so much water. Corn plants out in the middle of the hot sun in like Kentucky or Kansas or whatever, just tons and tons of corn just out there in a big dry field, and it's fine because it's a C4 plant. It's got this layer between the air intake and where the CO2 is actually, the CO2 in the water is actually getting used. So that's a C4 plant. Cam plants are a little bit different. It's in the same cell. It's in the bundle sheath cell, but it takes in CO2 at night. And turns it into malate, this malic acid. And then during the daytime, the malate can get turned back into CO2. So it's still kind of a C4 system. It's just a different C4 system because it's a 4-carbon thing. So they can get 4 CO2s out of every malate, and they run the Calvin cycle off of that that stored malate. So the CO2 goes in, and they go through. And the malate actually turns into pyruvate. So actually, I got that wrong. Sorry, they don't get 4 CO2s off of malate. They get 1 CO2 off of malate, and it turns into pyruvate, which can then also form starch and sucrose. Remember, it's part of glycolysis. So yeah, sorry, my bad. Self-edit. The malate, 1 CO2 comes off, the other 3 end up as pyruvate, and that can help form the glucose. But yeah, all of them have the Calvin cycle, all of them have RuBisCO. It's just how the CO2 actually gets into the Calvin cycle. This one separates the intake from the air from the input to the cycle. This one separates it by time by storing it as malate during the night. That way it opens its stomata at night to pull in the CO2 and push out the oxygen, and then it can keep them closed during the day. So you can imagine that a lot of desert plants will use this method, cactuses and stuff like that. Excuse me. Now, rice is the most popular grain in the world. Just think about China and Japan and Taiwan and all of those places, Indonesia, lots and lots of rice. It's a C3 plant, so it can't handle drought very well. So what they're trying to do is the C4 rice project. They're trying to turn rice into a C4 plant, to genetically modify rice to give it that insulating layer. And then that way the rice can grow in drier conditions. It can grow in places it could never grow before. You can grow more food, and you can feed the world, right? Imagine being able to grow rice in Africa. That's kind of the goal of the C4 project. Being able to grow rice in conditions that natural rice would just never be able to make it. So that's C3 versus C4 versus CAM. You have to know the difference between the 3 in terms of strategy. C3 is the simplest one, know nothing. C4 has that barrier. There's a cell that kind of intercepts the CO2 and then processes it as a carbon molecule that can get passed along instead of a gas. And then that way they can have a barrier. And then CAM photosynthesis is daytime, nighttime. a daytime nighttime thing. So turn the CO2 into malate during the night, and then turn the malate into CO2 during the day, and pyruvate as well, and then that can help out the Calvin cycle to make sugar. That brings us to the last piece here, so I'm going to pause and redo the thing. Okay, last piece here, kind of a short video today, but that's fine. Fixing carbon fixation is what I've called this one, and this is about the inefficiency of RuBisCO. So take that away. So we're going to fix carbon fixation. This is carbon fixation. CO2 comes in with RuBP. RuBisCO puts it, rearranges it, makes it into two of these 3PGs. And then that goes on to make, the triose phosphate, the G3P that gets pulled out as sugar. So sugar comes off of this side. Let's actually add that in, right? Sugar, yay! We want that. We want sugar. We want that side of the cycle. But RuBisCO is it's a great enzyme. It fixes carbon, but sometimes it does the wrong thing and fixes oxygen instead. It takes oxygen and adds it to the RuBP instead of carbon dioxide. So you can't make two 3PGs because you're missing a carbon. And instead it makes one of the 3PGs, but the other one becomes phosphoglycolate, which is not 3PG and thus can't go and make sugar. This happens about 25% of the time. So it does the good thing 75% of the time, does the bad thing 25% of the time. This one can go off, but this one needs some processing. And what has evolved in plants is that this one gets turned into glycine, which it attaches to the glycine and the amino acid glycine, and then it gets turned into serine, then glycerate, and then it gets turned into 3PG, but that 3PG turns into RuBP instead of going over this way. So this is a way for the plant to recycle it back to RuBP, so it can at least maybe hopefully go through this part of the cycle. But in this process, because it's using an amino acid, it has to kick off the NH3, and NH3 is toxic. It's also kicking out the CO2. It's actually taking out the CO2, and hopefully that CO2 can come in and do this side of things. So yeah, it also uses ATP. Just a bad thing in general. This is bad, right? It can't make G3P. It needs to reprocess back to RuBP. The reprocessing releases ammonia, which is toxic to the plant, and it uses energy. It would rather use that energy to make sugar instead of just wasting it through this cycle. But this happens 25% of the time. And you're saying, hey, Mr. B, photosynthesis evolved like billions of years ago. Why the heck hasn't it been fixed? Well, I guess this is just good enough. And there hasn't been anything that has evolved a better version of RuBisCO that doesn't make this mistake. Maybe something about the chemistry is just fundamentally means that oxygen can come in and kind of sneak over. Remember, oxygen is very, very good at stealing electrons. So yeah, this is that's the bad side of the cycle, and we try to avoid that, but you kind of can't. So what we are working on is genetically modifying these plants to have a better reprocessing method. So trying to take this 2P glycolate and somehow turn it into the 3PG without going through the amino acids, or maybe turning it back into RuBP without going through the amino acids, so that at least we can avoid the extra energy wasting and the extra NH3 waste and the CO2 coming out, which would just kind of go back out into the air and stuff like that. If we can avoid this piece with some kind of different molecular method, that would be great. And that's what they're working on. And they've identified it. They've actually made some plants that do it correctly. It's very, very early stages research, but you might hear about super plants at some point. And this is the actual article from the Frontiers in Plant Science journal, March 1st, 2018. So about 2 years ago they were working on this. And then this is an Ars Technica kind of general science article about it. And I'll have links to both of these down in the description, as always. That's the end. That's the end of our content. It was a 25-minute video this time around. It was kind of a quick one. So we've talked about the light spectrum and grow lights and how they need specific spikes. You saw that CD, the CD demonstration from my fish tank. I talked about the photomorphology affecting a plant's actual structure based on what kind of light we give it. Then we talked about the different kinds of carbon cycles. C3, which is just your normal Calvin cycle. CO2 comes directly in. C4, where the CO2 comes in, gets turned into a 4-carbon molecule, which then passes the CO2 off. That way you can have separation between the stoma and where the water is actually being used. And then CAM, which separates it by time. I like to remember that one by the AM in CAM, like morning and night. CAM, kind of a random useful way to remember it. So yeah, that's it. That was Monday's video, and thanks for listening, and I'll see you in class.